Hello and welcome to Biomentors. Today we are going to look upon and discuss about auxin, and this would be part 2 of our plant hormone lecture series. So let's get started. In the last lecture we discussed about auxin discovery, what are plant hormones, auxin biosynthetic pathway, its bioassay, as well as its types. Today we are going to study about the polar transport in auxin, the functions of auxin, as well as the auxin signaling pathway. So let's get started. So now, we will look upon the polar transport of auxin. So auxin is produced at the apical meristem. As we know that auxin is always produced at the apical meristem, either shoot meristem or in the root meristem. Auxin moves from one cell to another cell by polar gradient, which means from tip it transports to the base. So the tip portion is called as distal end and the basal end is called as proximal portion. It moves from distal to proximal end, which is mentioned. This is why cutting roots at the base, which means cutting root at the proximal end of the stem, promotes rooting. Okay. Now auxin transport proteins are located at the base of the parenchymatous cells, and transport is not sensitive to gravity and always moves in a polar direction. So auxin transport is always unidirectional and it is not affected by gravity, or rather it is referred to as gravity independent, from shoot to root, from proximal part of root to the distal part of root. From the distal to proximal part of the root and from the distal to proximal part of the shoot. Auxin are transported and it is always unidirectional. They can be transported into two pathways such as apoplast and symplast. Which is apoplast, which means a pathway with the help of its the auxin does not enters into the cell. And in the symplast, it is through the plasmodesmata the auxin is transported. So there are two pathways. Now we will see what are the transporters for auxin. Auxin transport from the distal to the basal portion of a plant. So there are two transporters, namely AUX1, this is AUX1, which is a symporter. This is referred to as symporter, which means it needs two molecules to enter inside a cell. Okay, it is sim means two. Okay, and pin transporter, which is located at the basal part of the cell, and AUX1 transporter is used for influx. यानी कि AUX1 जो ट्रांसपोर्टर होते हैं वो इनफ्लक्स मतलब ऑक्सिन को अंदर लेते हैं सेल्स में, और जो पिन ट्रांसपोर्टर है वो इफ्लक्स मींस exit of auxin outside the cell. Now auxin are of two types. Either it is IAA minus plus a free plus H plus ion, a hydronium ion, which is referred to as deprotonated form, and this form is seen at pH 7. When the auxin is in deprotonated form, it generally travels by symplast pathway. Okay, it is traveled by symplast pathway through the plasmodesmata. And if auxin is in deprotonated form, then it is transported by apoplastic pathway. And remember, the auxin, the protonated form of auxin, is lipophilic in nature, which means it loves lipid, so it can pass through the plasma membrane. Sorry. Now, as we have seen here that there are two types of auxin, either protonated or deprotonated. So the protonated path follows apoplastic pathway. So in this figure, this is a deprotonated form of auxin, right? The deprotonated form of auxin, and here is the protonated form of auxin. So protonated form of auxin can pass easily into the cell's cytoplasm, but deprotonated form needs energy to pass through this. Protonated form is lipophilic, and thus auxin can enter into them easily, while the deprotonated form is not lipophilic, and thus it requires a symporter, which is Auxin symporter. Location is at the plasma membrane and helps in influx, which means it brings auxin inside the cell, which is referred to here as influx, Auxin influx carrier. This PGP transporter is also there. Now, which is spin transporter? We refer to as spin transporter. So this transporter is for efflux, which means auxin exists out from this part of the cell, spin transporter. Now, when auxin enters into the cytoplasm of the cell, where the pH is 7, the protonated form converts into a deprotonated form because of the pH, and H plus ion increases in the cell's cytoplasm, and thus there is another transporter, which is another pump, which pumps out H plus ion outside the cells, thus creating a higher concentration gradient inside the cytoplasm of inside the cell. Okay. When pin accepts IAA and enters into the, now when the pin accepts IAA minus and enters again into the apoplastic pathway, which we have seen that it enters apoplastic pathway again at pH 5 or pH 6. Then again it enters symplastic pathway via Auxin receptor, and then finally to the growing parts of the plant they are transported. Okay. Now this is an important thing to note. I forgot to mention that when auxin exit from the cytoplasmic region, an expansive protein, expansive protein is activated, and this protein leads to elongation of cells, and thus it is also a function of auxin to promote cell elongation, and with the help of this proton gradient, expansive protein activates and elongates the cell. So this was the polar transport of auxin. The main two transporters, Auxin and pin transporter, which is for influx and efflux. And there are two forms of auxin, deprotonated and protonated form. Now, role of auxin. The role of auxin in the plant growth and development is for rooting. It promotes adventitious rooting. Then vascular inhibition and partnering. Vascular initiation. So auxin initiates cell differentiation in xylem and phloem cells. If high concentration of auxin is seen, then it promotes xylem. If high concentration of auxin is there, then it promotes xylem. If low concentration of auxin is there, then it promotes phloem. So this is the vascular differentiation. Then it induces flower development, embryogenesis, and tropic movements like phototropic movement is caused due to auxin. Now the most important part of this is auxin promotes elongation of cells and tissues. As we see that auxin promotes formation of expansin protein, and that leads to cell elongation. So here is the control series where water for 18 hours is given, the control series without auxin. And if we add auxin to this solution, then we can see elongated cells. Auxin promotes rooting. So auxin is very good rooting media for cuttings, and it promotes adventitious rooting in such cuttings. So this is the control series, and this is IAA induced or IBA induced series, and we can see rooting in this plant. Promotes apical dominance. So auxin also promotes apical dominance, and this apical dominance is a phenomena where the apical portion of the plant continuously increases and the lateral growth of the plant is suppressed due to this. And we will see why the apical dominance is of great importance in our next slide. It also prevents leaf abscission, as we have discussed in our previous video that it prevents leaf abscission because it is a negative regulator of senescence, prevents or delays aging in plants. Same, induction of callus growth. So when auxin and cytokinin are equal in proportion, then it induces callus growth, enhances fruit growth. In certain plant, auxin also enhances fruit growth. For example, pineapples. It also promotes parthenocarpic response in some plants. Auxin forms the developing seeds, which results in the fruit growth. Now, auxin and apical dominance. So here is the diagram of a plant. Now, let me to take a laser pointer. So auxin induces apical dominance. So due to presence of this apical bird, there is auxin inside this apical bird, and that's why the lateral bird remains inhibited. So the growth of lateral bird remains inhibited and the plant grows vertically. Okay. Now, if the apical bird is removed, auxin is less produced, which is not produced, and lateral bird grows, and thus the plant gets bushier. So auxin present. If auxin is present, then it causes apical dominance in the plant. And if auxin is absent, then it causes lateral growth only when the auxin is removed. Okay, so this was about the apical dominance. Now commercial uses of auxin. So 2,4-D causes a plant to grow itself to death, which means it is used as a herbicide or weedicide. As shown in the figure in the control series, no auxin was given, but in this 2,4-D is given, and some weeds are dead, more rapidly absorbed by broad levels of plant, and most often weed and feed lawn fertilizers. They are absorbed and they causes death. They causes senescence in such type of plants, and thus it is used as commercial herbicides. It also stimulates adventitious rooting, as we have discussed, sprout prevention in pruned trees, fruit thinning, and fruit holding depending on the stage of the development. So there is an important point I forgot to mention in this part of the that Auxin produces another hormone which is called as strigolactone. Auxin produces this hormone, and this hormone then induces the formation of BCR, sorry for the inconvenience. I forgot to mention this portion. So it promotes, then it expresses BRC gene, which is branch 1 gene, and this inhibits the lateral bud formation. And with the help of strigolactone, Auxin induces apical dominance in a plant. But when Auxin is removed, no strigolactone is produced and no BCR gene is produced, and thus lateral bud is not inhibited, and we can see the lateral growth in the plant. So this was all about the uses of Auxin, and now we are coming to the signaling pathway of Auxin. So Auxin follows a signaling pathway which is 26S proteolysis system, which means that the signaling pathway has ubiquitin proteins and which degrades the inhibitor. So what are the key players of signaling pathway? The first player is the receptor, which is TIR. The full form of TIR is transport inhibitor response. Then inhibitor, which is Auxin IAA. It is referred to as Auxin IAA. The ligand is itself Auxin, and ARF, which is the transcription factor, and the full form of ARF is Auxin response factor. Now, initially, when Auxin is not present inside the cell, then ARF and AUXIAA always are together. And because ARF and AUX, which is AUXIA, is an inhibitor, and thus ARF do not have a free site, and thus transcription is not seen when the Auxin is present. All right, so now we will see the signaling pathway. In absence of Auxin, I have mentioned this earlier, that AUXIA binds to the binds and suppresses the transcriptional activity of ARFs, which is seen in this figure. It suppresses the activity of ARF. When Auxin is present in form of molecular glue, so Auxin binds, Auxin first binds with TIR1, which is transport inhibitor response. Now this TIR1 has ubiquitin, and why it has ubiquitin? Because it is made up of 3 types of things. The first is F-box, and the second is choline, F-box, and SCFs. So now F-box contains ubiquitins, and which causes ubiquitination of inhibitors. Thus, ubiquitination, which means that ubiquitin is a small protein which binds to other protein and causes their degradation. So ubiquitin binds to the inhibitor and degrades the inhibitor. Okay, and AUXIAs are degraded, and the repressor is removed. The repressor is removed and ARF are then free to bind to DNA and cause changes in the transcription. And then finally ARF, transcription factors are free, and thus transcription of, again the transcription is done, DNA and important proteins are formed. And it is important to note that the transcription of TIRs, TIR AUXIA, which is also a repressor, is also done itself by transcription factor. Remember, it also transcribes ARF. Okay. Now, this was all about auxin signaling. In our next lecture, we will talk about another hormone, which is cytokinin, and also talk about gibberellins. So if you are watching this video, do give this video a big thumbs up and subscribe to our channel if it is helpful for you. Thanks for watching.